In the digestive canal, the master of absorption is small intestine. About 90% of absorption of all nutrients happens in the small intestine. Recall, the lining of the small intestine is mucous membrane, and when we say absorption, we simply mean nutrients from lumen, the space that we have inside the small intestine, pass through the mucous membrane and eventually get into the capillaries that we have below the mucous membrane. So here we're looking at one of the epithelial cells that we find in the mucous membrane that lines the small intestine. Because this cell is expert in absorbing nutrients, we refer to it as absorptive cell. As you can see, the absorptive cell does have microvilli. Microvilli increase the surface area of the cell to increase absorption. And we see that the absorptive cell, which is an epithelial cell, through hemidesmosome junctions, is attached to the basement membrane. And then below the basement membrane, we can see a connective tissue. And in the connective tissue, we're looking for the capillaries that were supposed to absorb nutrients into these capillaries. For example, in this image, we can see a fenestrated blood capillary having these pores, these fenestrations, allow this blood capillary to be more permeable and absorb more nutrients. The free surface of the absorptive cell, the surface that is not attached to anything else, basically it faces the lumen of the small intestine, is referred to as apical surface. But the surface that is attached to the basement membrane is referred to as basal surface. And when we analyze the steps of absorption, first the nutrient is in the lumen of the small intestine. Then the nutrients must pass through the apical surface, get inside the absorptive cell, and then the nutrient has to pass through the basal surface in order to be absorbed into the blood capillary. So absorption requires two steps. First passing through apical surface, then passing through basal surface. And we start with a monosaccharide named fructose. As you can see here, we have one fructose molecule in the lumen of the small intestine. If we break down sucrose, which is a disaccharide, we end up with a fructose and a glucose. And in this video, I'm going to explain how we absorb these two monosaccharides. To absorb fructose, when we analyze the apical surface and the basal surface of the absorptive cell, we notice in both surfaces we have some proteins that they're capable of transporting fructose. We name these proteins transporters. And then when we follow their actions, we see that each one of these transporters, without breaking down ATP, transports fructose. And we know that this type of passive transport is called diffusion. So absorption of fructose is through diffusion, and we use these transporter molecules to facilitate the diffusion. In short, we can say that fructose is absorbed through facilitated diffusion in both apical surface and basal surface, which that simply means absorption of fructose is entirely passive. We don't need to use any breaking down ATP in order to absorb fructose. Now the question is, how about absorption of glucose, which is a very important major monosaccharide in our body? When we follow the absorption of glucose, quickly we notice that its absorption is slightly different, because when we look at the apical surface of the absorptive cell, instead of a transporter, we see a protein that is called symporter. Recall, symporter is a protein that transports two different things in the same direction. It simply means that this protein absorbs glucose as long as it absorbs a different type of nutrient. And absorption by a symporter happens in the same direction. In this case, the symporter is going to take glucose and the other nutrient both inside the cell. When we follow this symporter, we see that the symporter is capable of absorbing sodium ion and glucose, but the ratio is important here. We see that 2 sodium ions and one glucose must bind to the symporter. So the ratio is 2:1. And then the symporter transports all of them inside the cell. And we know whenever we use symporter, that's an active transport that is referred to as secondary active transport. So we say absorption of glucose and sodium ion into the absorptive cell is by secondary active transport using a symporter. And then when we get inside the cell, we see that sodium ion and glucose follow different separate journeys to be absorbed through the basal surface. First, we start with glucose. We see that on the basal surface of this absorptive cell, we have a transporter that without ATP hydrolysis does the facilitated diffusion and transports glucose outside the cell into the blood capillary. However, to absorb sodium ion into this blood capillary, on the basal surface, we need a protein that is called sodium ion pump. The term pump tells you that this protein has to break down ATP, use the energy that is released from ATP hydrolysis to transport, to push sodium ion. So that would be a primary active transport done by the sodium ion pump. So in short, on the basal surface, absorption of glucose is passive, but absorption of sodium ion is active. Another monosaccharide that its absorption is exactly same as glucose is galactose. Recall that we get galactose, for example, by breaking down a disaccharide named lactose. When you break down a lactose molecule, you get a glucose and a galactose. Galactose competes to bind to this symporter with glucose. So in general, if you imagine that here we have a galactose molecule, the symporter can actually allow that galactose to bind to it, but recall that we also need two sodium ions to bind to the symporter, then symporter transport all of them inside the cell. So absorption of galactose inside the cell is by secondary active transport done by the symporter, and then inside the cell, galactose can be transported through the basal surface by facilitated diffusion. When I look at the entire image, I can see that absorption of fructose is entirely passive, absorption of sodium ion is entirely active. However, when it comes to glucose or galactose, they are absorbed inside the absorptive cell by an active transport, but they are absorbed through the basal surface by a passive facilitated diffusion.